Welcome to a new series of presentations to the scientific PowerPoints.
Today we will discuss about the role of matrix metalloproteases, or MMPs, in atherosclerosis. The learning outcomes of this presentation will be about the matrix metalloproteases' role in atherosclerosis, history of MMPs, their classification, their function and structure, their catalytic mechanism, their gene transcriptional regulation, their interactions with TIMPs, pathologies associated with MMPs, and their pharmacology. Atherosclerosis. In atherosclerosis, the artery becomes thicker, harder, and less elastic because of the buildup of a plaque called atheromatous plaque in endothelium tunica intima of the vessel. This plaque can be damaged by chemicals, for example by smoking, high levels of blood pressure, high levels of LDL or low-density lipoproteins. Because oxidized LDL enter endothelial wall via the transporter like VCAM1. Then the macrophages enter endothelial wall via the transporter like VCAM1. And after they engulf the oxidized LDL, the dead macrophages are the foam cells which are created. Then the relish of cytokines, MCP1, take place, and IFN gamma or else interferon gamma and TNF or else tumor necrosis factor in more mature plaques. As a consequence, more monocytes recruited in the era. This way a looping circle is taking place, and as a consequence a fatty streak is created, which is thrombogenic, or with other words site, which can create blood clotting. Furthermore, the muscle layer secretes calcium in the fatty streak, and the LDL deposit them as calcium crystals, which can be removed by HDL. Those crystals stiffen the arteries, and the recruitment of platelets on the plaque cause relish of PDGF or else platelet-derived growth factor, which encourage growth and migration of smooth muscle cells that is assisted by MMPs. These smooth muscle cells migrate from the tunica media to the tunica intima. Then those smooth muscles secrete collagen, proteoglycan, and elastin fibrous cells, which form the fibrous cap around and over fatty streak. The fibrous cap and the fatty streak are the atheromatous plaque. The fibrous cap can rupture, and as consequence the foam cells can enter the bloodstream that can form blood clots. When the clot is around 70%, it can cause artery occlusion. As a result, the blood flow is decreased, that causes ischemic conditions, which cause downstream cell injury, cell death, and weaken vessel wall, which can cause abdominal aortic aneurysms. Also, the plaque rupture can cause embolism, or with other words blockage of small vessels, that can cause acute myocardial infarction and stroke. Atherosclerotic plaques are vulnerable and more prone to rupture when they characterized by thin collagen caps, and when they contain increased levels of proteases, including MMPs. Now let's talk about the initiation of collagen breakdown in those atheromatous plaques. It requires interstitial collagenases, which includes an MMP subfamily, including the MMP1, 8, and 13. The MMP1 and 13 overexpressed in both hyperplastic and hyaline type of atherosclerosis, while the MMP8, which is known as neutrophil collagenase, overexpressed only slightly, while also neutrophils recently reported in atherosclerotic plaques. Let's talk now about the role of MMPs in atherosclerosis. The MMP1, 2, 3, 8, and 9 involved in vascular remodeling by degrading the matrix and separating the tunica intima from the media, which allows smooth muscle cell migration, proliferation, and accumulation of monocytes that later on lead to occlusion of the vessel, and as a consequence cause ischemic events to tissues in need of nutrients. Also, MMP10 acts as clot destroyer in atherosclerosis, attack fibrinogen and prevent its conversion to fibrin, which allow the netting of platelets that lead to plaque formation, and later on vessel occlusion. Homocysteine, psi, increase the likelihood of endothelial cell injury, as can be seen in hyperhomocysteinemia. Increase the inoxidative stress, which as a consequence activate MMPs, also acting as a signaling molecule on peroxisome proliferator activated receptor gamma, and on N-methyl D-aspartate or NMDA receptor. Nitric oxide is beneficial in some cases for deactivating MMPs. In other cases, it's shown to be harmful. Further studies investigate scenarios of beneficial versus destructive. H2S, hydrogen sulfide, decrease the activities of MMP by acting as antioxidant, and also it is a vasodilator. Also in atherosclerosis, increased levels of C-reactive protein in blood are noted that may indicate likelihood of atherosclerosis. In atherosclerosis, there are no symptoms until significant blockage take place. When this significant blockage occur, symptoms such as stroke-like symptoms, angina, or with other words chest pain, high blood pressure and renal failure, and leg pain can occur. Risk factors for atherosclerosis are: with increased age, the risk also increases. If there is family history record, and also males and postmenopausal females, while decreased risk in premenopausal females are noted because of the protective effect of the estrogens. Treatments are lifestyle changes such as smoking cessation, diet, and exercise, surgery such as angioplasty and stent, endarterectomy, and bypass, medication such as cholesterol medication such as statins, antiplatelet medication such as aspirin, blood pressure medication such as beta blockers, diuretics, and ACE inhibitors. And now let's talk about the history of MMPs. They first described in vertebrates in 1962 by Jerome Gross and Charles Lapierre. By observation of MMP's one enzymatic activity, the collagen triple helix degradation, by placing a tadpole tail in a collagen matrix plate. Therefore, it named interstitial collagenase MMP1. Later on in 1968, it purified from human skin. Classification: MMPs are endopeptidases. They belong to the superfamily Metsinsin, which they depend on zinc cation for their catalytic activity. MMP family has at least 26 known members that are grouped according to their substrate specificity. Collagenases MMP1, 8 and 13 degrade fibrillar forms of collagen. Gelatinases MMP2, 9 specific for denatured collagens and collagen type 4. Stromalysins MMP3, 10 and 11 cleave non-collagen components of extracellular matrix. MMPs MMP14, 15, 16, 17 and 24 are membrane type MMPs. Metalloproteases are the MMP7 and 26. Metalloelastase is the MMP12. Enamylysin is the MMP20. While Metsinsin superfamily also contains adenomyosins, cell surface, transmembrane proteins, and ADAMTs, which are multi-domain extracellular proteases. Function: they cause pericellular proteolysis, in other words, they degrading extracellular matrix proteins in order to freeing space for cell migration. Also, they cause cleavage of cell surface receptors, release of apoptotic ligands such as the Fos ligand, inactivation or cleavage of Fos ligand, which case apoptosis that observed in cancer. Also, inactivate chemokines and cytokines. By cleaving them, they make them truncated, which cannot bind to their receptor. That consequent as no movement of lymphocytes. This way, no inflammation is caused. No movement of cells during development by CXCLs. No activation and release of cryptic bioactivations and trapped hormones from the extracellular matrix. Release and cleavage of growth factors and cytokines, such as membrane-bounded, for instance, EGF, epidermal growth factors, and from the extracellular matrix ligands such as the transforming growth factor beta, TGF-beta ligands release. Release of growth factors from binding proteins such as insulin-like growth factor, IGF from IGF binding proteins, which lead to the activation of growth factors. MMPs also modifying the activity of signaling molecules. Modifying interactions and cleavage between cell adhesion molecules chems, between cell to cell. Modifying interactions between chems with components of extracellular matrix, cell to extracellular matrix. Also, they play a role on migration, angiogenesis, apoptosis, host defense, cell proliferation, differentiation, development, embryogenesis, and morphogenesis, for example, the webbing of the fingers, or with other words, the separation of the embryo's fingers, may affect leukocytes' entry into the central nervous system, can allow blood cells and monocytes pass the extracellular matrix and cross through the blood-brain barrier. MMPs function in tissue remodeling, wound healing. 1. They play a role in the removal of damage at the extracellular matrix. 2. The degradation of capillaries, their basement membrane for angiogenesis. Angiogenesis stimulated by VEGF or vascular endothelial growth factors, which activate VEGFR receptor of nearby vessels. 3. The migration of epithelial cells, their adhesion and dispersion. Angiogenesis stimulation in endothelial cells of blood vessels. The VEGFR1 receptors have as stimulating ligands the VEGF-A, VEGF-B, and PIGF, while the VEGFR2 receptors have as stimulating ligands the VEGF-A, VEGF-C, and VEGF-D. Those two receptors are the most important for angiogenesis. while in endothelia of lymphatic vessels. The VEGFR3 receptors have as stimulating ligands the VEGFC and VEGFD. The lymphatic vessels are major route of metastasis. After the receptors bind with their ligands, that cause receptor dimerization, then receptor autophosphorylation, then the signaling pathway cascade goes further down, which lead to the phenomenon of angiogenesis. Now let's see the structure of MMPs. In this image we see the three-dimensional structure of MMP9 complex with an inhibitor. The green spheres represent the calcium cation molecules, while the gray spheres represent the zinc cation molecules. Also, MMPs have four common domains: propeptide domain, catalytic domain, which has the zinc molecule, Hinge region, and hemopexin-like C-terminal domain. Propeptide domain is 16 to 19 residues long, is a globular prodomain, contains a part of cysteine switch, which contains a conserved cysteine residue. This switch interacts with zinc inactive site, which prevents binding and cleavage of a substrate. MMPs initially synthesized as inactive zymogens, which are a latent form, secreted as pro-MMP, which are proenzymes, which has a propeptide domain. The propeptide must be removed in order to cause enzyme activation, which requires extracellular activation by proteases, such as trypsin, plasmin, plasminogen activators, elastase, and other MMPs, or intracellularly activation, such as by furin, that cause disruption of bond between cysteine residue in pro-domain from zinc ion in catalytic domain. This way is facilitated the removal of propeptide, which transform the pro-MMP to its active MMP form. And here we can see some examples of extracellular activation of pro-MMPs to active MMPs. And here we can see some examples of intracellular activation of pro-MMPs to active MMPs. And now let's talk about the catalytic domain. X-ray crystallographic data shown that this domain has flattened shape measuring 35 to 30 to 30 Armstrong, 3.5 to 3 to 3 nanometers. The active site is 20 Armstrong, 2 nanometers groove. There is a catalytically important zinc cation in this domain, is bound by 3 histidine residues found in the conserved sequence HXXHXXGXXH, which is a zinc binding motif. And now let's talk about the Hinge region. It connects the catalytic domain to the C-terminal domain, is up to 75 amino acids long, and has no determinable structure. And now let's talk about the hemopexin-like C-terminal domain. It has structural similarities to hemopexin, serum protein. That's the reason that it's called hemopexin-like. The Hinge region link it with the catalytic domain. It has four-bladed beta propeller structure, is a large, flat surface involved in protein-protein interactions, which determines its substrate specificity. Also is a site for interaction with TIMS, tissue inhibitor of metalloproteases. And now let's talk about the transmembrane GPI anchoring domain, which anchors NTMPs, membrane-type MMPs, to plasma membrane. And now let's explain its catalytic mechanism. In the active site, there is a metal atom, usually zinc 2 plus cation, that interacts with water's oxygen, which is partially negative, which creates a zinc-oxygen bond formation, while a nearby residue, usually glutamate or aspartate, acts as a base that pulls the hydrogen away from water's oxygen. That lead to the creation of a hydroxy that is a better nucleophile. This way enable it to hydrolyze the peptide bond, increase a rate of reaction. When hydroxy attacking the carbon atom of the electrophile carbonyl of substrate, it forms an tetrahedral intermediate, which collapses in series of steps, or with other words, it breaks up, and then rehydrolyze the peptide bond. Their gene transcription is induced by soluble factors, including cytokines, growth factors, integrin signaling by cell-cell and cell extracellular matrix interactions. Also, their transcription dependent on the binding of AP1 activator protein 1, formation of heterodimers, CFOs and C-jun to the AP1 site, which lead to maximal activation of the inducible MMPs promoters of MMP1, 3, 7, 9, 10, 12, and 13. TIMS, or else tissue inhibitor of metalloproteases. are smaller, 20 to 30 kilodalton, naturally occurring proteins. They are TIMP1, 2, 3, and 4, each with its own physiologic role. TIMP1, 2, and 4 found in a soluble form. TIMP3 is anchored to the extracellular matrix. They binding and inactivate active forms of MMPs by forming non-covalent bonds with latent and active MMPs. This way they control the breakdown of extracellular matrix components. Also, they can undergo changes in their molecular dynamics, which induced by their interactions with proteases. They can have an effect on cell growth, differentiation, migration, anti-angiogenesis, anti- and pro-apoptosis, and synaptic plasticity. Also, other MMP inhibitors are the TFPI2 and REC. And here we can see the NT1 MMP complex with TIMP. The green spheres represent the calcium ions, while the gray spheres represent the zinc ions. Pathology. MMPs are important biomarker of disease. They have important role in cancer. Tumors have increased activity of MMPs. 1. They provide them space by degrading the extracellular matrix. 2. They trigger angiogenesis. 3. Cause failure of cell death, for example, by inactivating the Fos ligand. 4. They degrade basement membrane that enable tumor invasion, metastasis, especially by MMP2, 9, and MTMMP1. Also, MTMMP1 expression correlates with malignancy of multiple tumor types, including lung, gastric, colon, breast, cervical carcinomas, gliomas, and melanomas. The acute and chronic cardiovascular diseases are characterized by dysregulation of balance between MMPs and TIMPs. In aortic aneurysm, excess MMPs degrade the structural proteins of aortic wall. In chronic wounds, excess MMPs cleave collagen fibroblasts, which produce tissue fibrosis. In rheumatoid arthritis, osteoarthritis, especially MMP1 play a role. Also, they have a role in congenital defects due to loss of differentiation. And also, they have a role in cirrhosis. Pathology. In the brain, MMP9, or gelatinase B, is induced during neuroinflammatory response. Is used by T lymphocytes to attack basal lamina layer of extracellular matrix. In this way, allow the crossing of blood-brain barrier. Its increased levels observed in allergic encephalomyelitis, NMS, or multiple sclerosis, more noticeably before acute attack. MMP2 and 9, or gelatinase A and B respectively, attack basal lamina that surround blood vessels. In this way, they altering their permeability. Their intracerebral injection opens the blood-brain barrier. Also, increased levels observed in CSF cerebrospinal fluid of MS patients that return to normal after steroid treatment, and in spinal cord wounds that lead to scar formation. Pharmacology. Synthetic inhibitors generally contain a chelating group, which binds tightly to the catalytic zinc atom inactive site of MMPs. Usually they interact with various binding pockets on the MMP of interest. This can making the inhibitor more or less specific for the given MMP. Pharmacological agents for MMPs are corticosteroids, aspirin for MMP2, doxycycline with trade name Periostat. This is the only MMP inhibitor that is widely available clinically. Is used for treatment of periodontal disease. Other agents are minocycline, tetracycline antibiotic. Rationally inhibitors are promising, such as Trolcade, Marimostat, and Cypamostat. Shown implications are toxicity, musculoskeletal toxicity, and failure to show the expected results. Also, collagen wound dressings keep MMPs busy with their collagen instead of degrading body's collagen, and provide collagen as a scaffold for the healing process.
And here we can see some useful references. The end. Like and subscribe. See you in the next one.